second. All right, so good morning, everyone, or good afternoon. Welcome to our Friday noon seminar. Today we have the pleasure of welcoming, hold on, pull up, Dr. Christopher Rowley. Chris Rowley is a postdoc currently at the McConnell Brain Imaging Center here in Montreal at the Neurological Institute at McGill University. He's working in the labs of Doctors Christine Tardif and Bruce Pike, working on developing more specific MR measurements for brain myelination. And he will be talking to us about that today. And I just wanted to mention as an aside, as well as being an MR expert, Chris is also a cycling expert. And for those of you who are cyclists in Montreal, correct me if I'm wrong, you have the second fastest time on the F1 track, something crazy like that.
Yeah, I got beat by a guy on a recumbent bike, so I don't know if that counts or not.
It still counts. So that's equally impressive. So I know your talk's focused on MR today, but if anyone has any cycling questions, you can also ask Chris. So with that, I'll give it to you. Thanks.
Thanks, Pete. And thanks everyone for coming. Today, I'm going to be discussing my work on using MRI to make inferences on the microstructure of the brain. Second, there we go. So for this talk, first I'm going to motivate my work with an introduction on the relationship between the structure and function of the brain. And then because we have a diverse group of interests here, I'll talk a little bit about why we might want to use MRI to study this. And then with that motivation, I'll get into some research that I've done previously on qualitative metrics to study brain microstructure. And then with a little bit of time left, get into some more quantitative and metrics with biophysical and physical modeling of the signal and tissue. So why might we want to study brain structures? So I took this loop from a talk from Nicholas Weisskopf that I really think encompasses brain plasticity and development. So if we start at the most basic portion and function, we might have a single input-output, such as listening to a tone or being able to move your leg. If we start to group these things together, we might move towards something like carrying on a conversation or playing hockey. As these functions become more ongoing and repetitive, we get towards a behavior. So you might notice the behavior of a Montreal Canadiens fan right now. There might be a little bit depressed behavior, harder to get a smile out of them. So that ongoing function leads to a behavior. And we know from classical neuroscience of Hebbian wiring, so neurons that fire together, wire together. So the initial structure of the neurons helps with the function, repetitive function or firing of neurons elicits changes in behavior, and that's reinforced with changes to the microstructure. So carrying forward with a microstructure, it allows us to initially create the function. So while you might need a cable and TV package to watch hockey, in order to view that in HD, you'll need a better TV and cable package to do that. If you want to be a better hockey player, you're going to need to more finely tune your neural circuitry to be able to do that. So with that in mind, it's probably best to appreciate this better if we look at the scales at which neuroscience work occurs over. So at the macro scale level, we can see the brain as a whole working on a scale of centimeters. We might see brain regions such as visual cortex at the back, motor cortex in the middle, and we might try to connect those two for something like hand motor or visual coordination. With MRI, we start to move towards the mesoscale. We can start to see separation of brain structures such as the white matter and gray matter. Now we're working towards millimeters. As we move higher up on the scale, we start to get towards histology. And with the histology, we can start to see the columnar processing units within the brain. So here we see a nice myelin stain and a cell body stain within the gray matter of the brain. Along with the columnar processing, we also see this layer-specific patterning within the gray matter of the brain, which has to do a little bit with the function of the region. And then as we get to the microscale or the nanometer resolution, we can start to see fine cell structures such as the tight wrappings of the myelin sheath around an axon, and even little structures within the axon itself. So just within this span, we can see there's 7 orders of magnitude between the macro scale and the microscale. MRI realistically will be limited by physical constraints towards the mesoscale in millimeters towards micrometers, but we can use biophysical modeling to try to bridge some of the gaps here. And that's one of the powerful reasons why we like MRI for neuroscience research. So how can we study this in vivo? In the quest to correlate structure with function, it would be best if we could probe the structure while the specimen is still functional. So MRI provides an excellent tool to do so with many contrast mechanisms at our disposal for studying the structures and composition of the brain. So if we compare it to other imaging modalities that are often used, why might we want to use MRI over something like X-ray or CT? Well, MRI in most imaging applications generates its signal from the water protons. So because we're getting our signal directly from the water, which makes up the soft tissue, we get good SNR from these tissue contrasts. This is in comparison to X-ray and CT, where we're really just visualizing the attenuation of X-rays, which is typically done through solid structures such as bone. So that's where you're getting the contrast, and it's very apparent in the X-ray. You're mainly seeing the skull and spine. You move towards CT, you can kind of see something's going on there. There's probably a brain, but that looks very windowed and leveled to be able to see that. Where MRI, that's very clear, there's a brain and you can see some structure within that brain. So another benefit of MRI is there's various ways to generate contrast within this soft tissue. I'll provide a brief overview of 2 mechanisms that pertain to the work I'll share today, but note there's several others that are frequently used. So one widely used contrast mechanism in MRI relates to the time or rate it takes for the magnetization to return to its thermal equilibrium state. So the rate at which this occurs over is R1. It's also known as 1 over T1 by many others. So this rate is strongly influenced by the main magnetic field that it's in, but also the local magnetic environment, which is dependent on macromolecules in iron and other magnetic ions. So because of this, it gives a strong tissue contrast, and you can use it in like one of 2 ways. You can acquire multiple images and fit this time curve, or you can select any point on this curve, optimize your sequence parameters for that to generate a T1 or R1 related contrast. If you do that, the image you get something like this, where this is an axial slice where we have the back of the brain towards the eyes, and you see really bright white matter, darker gray matter, and the CSF you can see in the ventricles is dark. So we can take this a step further if we're interested in looking only at the gray matter, which is kind of hard to parse out of a volumetric image, and display this on a cortical surface. I'll get into this a little bit more, why we might be interested in doing this, but it really helps us differentiate brain regions where we can see the bright primary regions such as motor cortex show up quite brightly and separate from association regions. Another contrast mechanism that I'm really interested in is magnetization transfer. And the way that there's a few ways to generate this contrast, but the way that my work aims to do that is by applying a radio frequency pulse that's at an off resonant frequency from the water resonant frequency. So by applying it offset to water, we are able to selectively excite the protons that are part of this bound pool because they're spinning at a slightly different frequency. So this will selectively saturate the magnetization of this pool, which undergoes rapid exchange with the water pool. If we then, after applying this pulse, excite the free water pool to acquire an image from the free water, we can indirectly view the impact of this pulse as a decrease in magnetization from the exchange in the free water pool. So if we do that and acquire 2 images, one with and one without this off resonance pulse, we can quantify the effect of this pulse. And if we map that across the brain, we get something like this. It does look quite similar to the R1 map. We see very bright white matter. We know there's a lot of macromolecular content, so the bound pool fraction is quite high. A little bit darker gray matter, we have more water there, so less macromolecules just due to the increased cell bodies. And we see no magnetization transfer in the ventricles and CSF because that's primarily water with no bound pool, ideally. And again, we can project this onto the cortical surface to look across the brain. So for surface-based analysis, how about would you do that? So starting off, we would take an MRI image for each subject, and we first need to segment where the gray matter and white matter is within the image. Once we have done that, there's a few software packages that can do that. Generally, you might need a couple manual touch-ups, but it's mostly computational. You feed each of these surfaces then into a volume preserving algorithm that generates a surface in the middle depth or any range in between the two. And then what you're really interested in is the MR signal. So this surface is based up of vertices. It's a triangulated surface. So you map the MR signal onto each of the vertices on the surface. And then you can get subject specific maps. So I think it's worth appreciating here that there's quite a bit of difference between just the shape of the brain between people. I know that surprised me a lot when I first started looking at MRIs. So if we're going to do a big study and compare between groups, we would generally like to be able to say, all right, this point on the brain, we want to be able to sample what this value is across everybody in the study. So in order to do that, you generally register these surfaces onto an average template, and then you have everyone's map in the same space. So now that we can do that, why might we actually want to do map surface values? So surface data extraction allows us to focus on the relevant data. So primarily my work has been focused on the cortex. In brain mapping, you have a big issue with multiple comparison corrections. So you're generally looking for ways to reduce the comparisons you're going to make. Otherwise, you get killed in the statistics. The differences in signal provide insight into the microstructure. So again, we have this T1 view is from the brain top down. We can see the motor and somatosensory primary regions showing up very bright, and the visual cortices bright, and the rest is a little bit darker. And we see this as pretty robust major findings in all of these contrasts that are reporting some sort of myelin content in some way. Same with MT-SAT, the quantitative R1 map, and a T1 divided by T2 weighted ratio. So this kind of myelin mapping seems to be robust, but we're slightly different to sensitive to slightly different things. And with that, what are we seeing with these contrasts? So some initial work was done by Bach et al. With some histology work in a marmoset. So this is a Myelin-St they did with the visual cortex, and this gets towards more motor regions. It's worth noting that the stain will be an inverse contrast. So the darker it is, the more myelin you see. So the really dark region is the white matter, and you have this nice band that goes around for V1 is characteristic of that region. And in this T1 weighted MRI, we see that band shows up again. So we have really good spatial correlation with this T1 contrast and a myelin stain. Another study a few years later by Stuber et al. Showed a little bit more by adding in a different contrast. So they looked at T2 star as well, in addition to T1, with a lipid stain and iron. So the ultimate conclusions of their study was that T1 was mainly sensitive to lipids and myelin content, and slightly sensitive to iron, where if you add this T2 star contrast, which is a dephasing of the spins, it's really sensitive to the local magnetic environment. So you have a very strong impact of iron on the signal. And it's probably most evident by looking at the stria of Gennari. So this is another visual cortex. It's the same thing that's in this image. It's very prominent in this T2 star image. It is there a bit in the T1 and lipids, but not as strongly as the T2. So ultimately, we know MRI can be sensitive to many biological parameters, but we can sort of tune our imaging sequences to increase our sensitivity to specific biological structures. So since we can do that, we have that at our disposal. From a biological perspective, why might we want to look at this MRI signal if it's going to tell us about myelin? So I think it's worth noting that despite all the computational processes occurring by the neurons, they're really outnumbered in the brain. So within the cortical gray matter, this is where the cell bodies are for neurons. They're outnumbered by a factor of 1.5 to glial cells, which make up the support network. And then in the white matter, this ratio goes up to over 15. So given the abundance of these cells, and they're in a space that's really limited by real estate, the skull encapsulates the brain. So the brain would like to grow as big as it can, but if we're limited by space, there must be some reason that they're there to justify their numbers and the space that they're taking up. Otherwise, the neurons could have it all to themselves. So the literature on the importance of myelin, and myelin's produced by oligodendrocytes, which is a type of glial cell. It's been long established, at least in classic neuroscience books, that the primary role of myelin is to increase the speed of signal travel down an axon, where it can go up to 10 times faster through saltatory conduction, where it's sort of jumping over the myelin sheath. And the first evidence of this was reported in 1925. So this has been a really long-lived field. More recent research has turned towards the metabolic support that these myelin sheaths can provide to the axons. So neural firing is very energetically expensive. So the myelin sheaths actually seem to be able to support these axons. And what you actually find is the axons that end up being the most myelinated tend to be the most active. So that's kind of cool. We're coming out, and potentially most interesting to us based on what I started with, the structure functional relationship, is we have this regionally distinct distribution of myelin patterns across the brain, and that's sort of how the myelin varies through the layers of the cortical gray matter. It's been used by many groups to parcellate the cortex, and the whole hypothesis behind this is that the structure of that region and how it's made up across the layers is built to a specific function. So if we can determine the structure of that region, we can have a little bit better understanding of the function of that region of the brain. So moving into some of the research and applying this, I'll begin by sharing some qualitative metric research that I've done. So starting with applying this in healthy aging, so using MR signal to see how the brain matures with age, and then applying that in a mental health case with bipolar disorder, and then looking at neurodegeneration with Huntington's disease. So it's been well established that the cortical gray matter in humans continues to develop well into adulthood, even though we feel like we're probably mature enough around 18. The brain thinks otherwise. So an initial study with over 1,500 subjects had showed, using this T1 divided by T2 weighted myelin metric, that by looking only between 18 and 35, we have a linear increase in this myelin correlated metric up to 35. So we're still increasing up to 35. Another group with a study of over 300 subjects showed this peak is actually probably between 40 and 60, depending on the brain region you're looking at, before we see a decline with neurodegeneration. So I think it's worth noting here that this is a multi-contrast metric. So we have increased iron sensitivity with this T2 weighted scan that's incorporated here, which I'll touch on in a bit. So what we were really interested in was, could we track this age trajectory with a smaller cohort? So the previous studies, we were 300 for one, over 1,500 at another. We'd really like to be able to apply this in clinical populations, and those study numbers are really becoming infeasible for many studies and populations. So we thought if we used optimized imaging, could we see the same thing with a smaller number of subjects? So we used some sequence optimizations from Bach et al., just that were optimized to get a T1 contrast within the cortical gray matter. We weren't really interested so much in gray matter, white matter contrast, which typical clinical scans might be optimized for. And also with this, we used the ratio approach to remove a large degree of hardware-induced inhomogeneity in the images. So what does that mean, or what does it look like? So on the farthest side to the left, we have our T1 weighted image. This is the contrast we're really interested in. But you can see there's some central darkening here, which is just shading induced from the hardware, just as a function of the coil placements and power disposition. So you can see it's really bright here, darker here, and a little bit brighter towards the outside. Because there's more going on, we can see it a little bit more effectively in here. So this was a proton density weighted image. We have very little tissue contrast. We're mainly using it to remove this bias field. And then when we take the division of the two, we get a much more homogeneous image. So the goal really of this is we want to be able to look at a point in this image and say, it means this in the anatomy, and it's not so much about the scanner hardware where we would have to look at it, and it would only mean something specific depending on where in the brain you were looking at. So moving this to surface, we have our images that we collected in 67 individuals between 17 to 45. So not quite a full age range, but a little bit more than that first study that we showed. We were hoping to capture the peak. If we look at these values across 3 cortical depths, looking from quarter depth being the furthest outside superficial to a deeper 3 cortical depth, we see these nice trajectories, and these were just 4 sample regions across the brain. Interestingly, we see good separation between the 3 cortical depths sampled, which I think speaks to the high contrast to noise ratio in our highly optimized imaging. That also took over 35 minutes to acquire these images, so it wasn't free to get that contrast to noise ratio. But because we were able to capture this peak, we wanted to fit it with a quadratic to be able to pull some information about this maturation. So we fit it with, we were able to reorganize the quadratic fit to extract the max value, K, and then the age at which the max occurs, which we're calling H here. So if we do this across the brain and plot those values, what we get are some nice maps. So just looking at the max signal that would theoretically occur based on our fit, we see a trend that we would expect. So the more superficial layers have less myelin, and as you move towards the white matter, it becomes brighter, which we assume correlates with a higher myelin content. What was more interesting to us was this temporal shift curve, or plots. So there was no statistical difference between the different depths, which suggests the different depths mature in terms of their myelin growth at a similar rate. So we wanted to take this one step further and use these H values as an input to a clustering algorithm to look at spatial distribution of maturation. So we put the H into a k-means clustering algorithm, and what was output was a pretty neat result. So we see some good spatial organization to this maturation pattern across the brain, with visual cortices and cingulate cortices maturing earliest at around the age of 34. And then association regions such as parietal and temporal lobes and frontal pole lobes peaking earliest. And then we also see this rostral to caudal development gradient across the frontal lobe, which matches a known functional gradient. So we're hypothesizing that the downstream target or the upstream targets have to myelinate to completion before the downstream targets. So what would happen in sort of a brain functioning state is you might have the idea to do something that's stored in a working memory portion, and then it works towards a concrete motor output, such as moving your arm or legs. So this area that's informing this area needs to be matured completely before the one after it does. So, and then we found the latest maturing region to be around 40. So it's a little bit earlier than the one study I showed earlier. So what are some implications from this work? We showed that with a high CNR, we were able to replicate trajectories with a much smaller cohort of subjects. And the T1 weighted signal in the cortex continues to increase to about 35-40. And this is what we're expecting. Some other histological work sort of supports this. Some earlier work from 1994 showed just a sampling of the cortex where they're hitting peak myelination at 45. I mean, this is pretty discretized, but it's certainly higher at 45 than it is at 60. And then some other work looking at myelin basic protein expression, which is a dominant protein seen in the myelin sheath. And then just in reference to the other study where they showed a later peak, this is tracking iron content in the brain with age. So the brain's actually really great at taking in iron. We need iron for a lot of metabolic processes. The problem is the brain isn't good at getting rid of iron. So once it has it, it's sort of stuck. And that's one of the problems with aging. So we're hypothesizing the earlier peak we're seeing here is because we're not as sensitive to this T2 contrast and iron loading that the T1 over T2 weighted contrast is sensitive to. And then the other implication of the study was the development seemed consistent across cortical depths. So this should really be revisited with some higher resolution imaging, but it's sufficient to say that at least at the resolution we're using at around 1 millimeter, we weren't able to detect differences, so we could simplify analysis for future studies. So because we had this worked out in healthy controls, we wanted to take it a step further, and we collaborated with a psychiatric group at McMaster University to look at bipolar disorder type 1. So a little bit about the pathology of bipolar disorder. It's characterized by cyclical episodes of depression and mania. So there's two types. Type 1, you experience more extreme episodes of mania, and type 2, a little bit less severe, sort of like diabetes with type 1 and type 2. It affects 1 to 3% of the population, with onset typically in the late teens to early 20s. And in the bipolar community, research community, there's arguments for it to be considered a developmental disorder, which makes it interesting to study with the developmental trajectories. What makes it more interesting to me is that there's myelin deficits reported in bipolar disorder with histological work. So this study here I highlighted is depression, bipolar, and schizophrenic groups here compared to controls, mainly interested in bipolar, but we see a significant decrease in the number of oligodendrocytes in bipolar disorder. So again, these are the cells that are creating the myelin sheaths. And another study also showed a decrease in glial cell density in the prefrontal cortex in bipolar disorder. So we have reason to believe that there could be an age correlation and myelin involved in the pathology of bipolar disorder. So we wanted to do similar to what we did in the healthy subjects, looking at trajectories. So for this plot, we've added a bipolar subject group, which is 40 individuals between 17 and 45. And the red plots are exactly what we saw before in the healthy controls. So we have a nice quadratic function here. But then the blue plot is the bipolar group. So what's very interesting here is it looks like a flattening of this trajectory across the brain. I wouldn't read too much into this separation here. It's likely due to just the restraint of the quadratic fitting more towards these data points. But definitely something's interesting is going on with the curve being more flat. So in order to look at that, we fit the signal as a function of age, squared age, and an intercept term. And then if we want to know about the curvature or the flattening of that trajectory, we can look at this coefficient here. So if we calculate that across the brain and see what that looks like, we get a map like this. So in the controls, it's what we expected. We have this strong inverted U shape that's based on this negative age coefficient. And then this bipolar disorder group, the coefficient really tends towards 0, which suggests if this goes to 0, you now lose the quadratic nature and the curvature within the plot and becomes linear. So that was a very interesting finding. And if we apply some significance to this, we see that if we remember back to the clustering results, that it primarily seems to be the later maturing groups that are predominantly affected by this. So they're potentially more susceptible to issues with this myelination trajectory. So if we want to relate this back, as a clinician, you might not be too concerned about changes in myelin levels if they don't have a functional outcome to them. So we correlated this with a cognitive score of verbal memory. So verbal memory seems to be pretty replicable in bipolar disorder as something that's seen in deficit with disease status. But we don't really expect changes in healthy controls. So when we did this correlation between the two groups, we found something interesting. So the gray areas are just the regions that weren't significant. So we're mainly looking at the colored in regions. But in controls, we found no correlation between our verbal memory scores and the T1 scoring of myelin content. So we know the T1 myelin content's changing over this time, so that because there's no actual functional change, there could be some sort of compensatory mechanism going on where changing myelin content is just preserving some level of function. Interestingly, though, there is a strong correlation once you look in bipolar disorder. So the higher the myelin score based on this T1 measurement, the better the verbal memory score is. So if the age trajectory with myelin's causing a decrease in your myelin levels, if you're more impacted by that, you're going to have a worse verbal memory score. So what we took from this study was the developmental trajectory appears to be altered in bipolar disorder type 1, and the signal correlated with their functional ability. And we're hoping to follow this up. It's currently being investigated with a longitudinal study by the same group at McMaster. So we're hoping to be able to look at a couple more variables. It's multi-institutional, more subject power, so we can look at more things like medication status as well. So up until this point, I've shown you results using optimized imaging that provides really good contrast to noise ratio, allowing us to see things in the cortex. But there's a large area of brain myelin imaging that uses this T1 divided by T2 weighted ratio. I think it's predominantly used because of its availability. These are two scans that are collected routinely for clinical purposes, just to what they're sensitive to. So finishing off my PhD, I was interested in taking advantage of this to investigate Huntington's disease, as there was a large multi-institutional study that was done called TRACKHD that had this data. So a little bit of background about Huntington's disease. It's a well-characterized disorder caused by a CAG trinucleotide expansion in the Huntington gene. It also has known increases in glial density in the cortex, which is known from histological studies. And what made it easy for me was there was a data set that already existed with 120 controls, 120 pre-HD, and 120 HD subjects. So what does this grouping mean? Well, the healthy controls did not have the sufficient trinucleotide expansion in their Huntington gene. And then this grouping of subjects with HD, they all had the expansion. And based on the expansion length, you can have an idea of the age at which symptom onset can occur. It's a very well characterized disorder. So once you have symptoms, then you're classified as having Huntington's disease, and there's stage 1, 2, and then it goes up from there. And then we separated the pre-HD group into A and B, depending on how far away they were from predicted diagnosis or symptom onset. So this will allow us to look at brain structure changes before symptom occurs and then right after. So again, the imaging, we're using this T1 divided by T2 weighted ratio. It's a multi-contrast myelin mapping technique proposed by Glasser in 2011. It's gained quite a bit of traction in the neuroimaging community. It's reported to have enhanced myelin-related contrast. I'm going to argue it's primarily due to the increased sensitivity of iron, which tends to spatially correlate with myelin content. But I think the huge benefit of it really at the time was the large removal of bias shading. So they gave this sort of simplistic equation where you have a T1 weighted contrast, which has myelin contrast and the B1 bias shading. And the T2 weighted contrast has inverse related myelin contrast, but still the same multiplicative bias shading. Whether it's the same or not, it's up for debate. But if you take this division, you end up with this sort of myelin squared or enhanced myelin contrast. And if you plot these results onto the cortex, they look pretty good. So people have ran with that. So looking at it in this specific data set, we're applying standard clinical images to this cortical myelin mapping process. It's worth noting that because they spent way less time collecting these images compared to the previous study, we definitely have a reduced signal to noise ratio in these images. It's quite evident here, but we do what we can, and it helps that they have a lot of participants in this study. So if we just look at the group average maps, here we've regressed out the effective age and imaging site before plotting the average values for each group. We had 112 controls after removing subjects that had issues with motion, 54 in the pre-HDA. So again, this is the furthest from symptom onset, 51 in Ph.D, and then with symptom onset, we have stage 1 at 68, and then 41 in stage 2, so a little bit more advanced. So if we look at the signal changes in the brain, we can see what we're plotting here is the coefficient from the general linear model in relationship to how the signal is changing relative to controls. So because we're already starting to see some yellow here, pre-HDA, we're already seeing the microstructures trending to change well before symptom onset. And this progressively increases well into symptom onset and with increasing severity. If we look at uncorrected p-values, they increase. And interesting to me was they sort of follow this distinct pattern, starting with the insular cortex and then spreading outwards. And because we have so many regions, I touched on it earlier, it's sort of an issue in neuroimaging. But once you do the correction for multiple comparisons, we're left with significant regions only in the later disease groups. So what can we take from this study? It's the first in vivo study of cortical composition in Huntington's disease, which is exciting. It could be reporting increased density of myelin or ferritin-loaded microglia. So ferritin is the protein that stores iron. So there's, again, the issues with this T1 divided by T2 weighted ratio. There could be a lot of stuff going on at the biological level that could be occurring. So the T1 has really good sensitivity to myelin. So things that could be changing are sheath thickness, the number of myelin sheaths, or even the length of the sheaths. But with this T2 weighted contrast, we have a high sensitivity to iron content. So you could have a decrease in the ratio of sheaths to oligodendrocytes because the oligodendrocytes store iron, an increase in oligodendrocyte ferritin, so iron loading in the oligodendrocytes, or because it's a neurodegenerative disorder, you could have this increase in ferritin-loaded microglia. So these are the immune cells of the brain, and they're just cleaning up the cells that are being degenerated. So I think this warrants for future studies in HD with a little bit more specific and higher SNR imaging to better study the cortical microstructure and see how it changes before symptom onset even better. So conclusions from this work. One of the benefits of qualitative MRI is it results in shorter scan times, potentially than quantitative metrics. It can be optimized to produce very high SNR and CNR, which will help you see differences across the brain that you might be interested in. And additionally, there's existing databases that may already have all the images you want to answer the research question that you have. These images are not cheap and they take a long time to get, so this is very helpful. Some of the problems, though, is they're difficult to standardize across sites. If you're looking at multi-institutional data, you could have different scaling and offsets just because of the hardware of the imaging used. Oftentimes, they can't even match the parameters across sites. MRI is a bit finicky that way. And again, as you introduce multi-contrast imaging like T1 divided by T2, you end up losing some of your microstructural specificity. So if we move away from that towards more quantitative mapping, we can see what we can do, spending just a little bit more time, potentially getting a physical constant that could be more standardized and potentially mean a little bit more. So in this case, I was interested in exercise-related cortical myelination. So there's a lot of recent work coming out showing that myelination is an activity-driven process. So the more active your neural circuits are, they need that metabolic support. So the myelin sheaths are thickening and growing around those axons. Again, we have the behavior modifying the microstructure, which affects your ability to function. So that's how I wanted to look at it. Just a quick review, R1 mapping. So we're looking at a physical quantity. This is the relaxation rate at which the magnetization returns to the thermal equilibrium. So it's governed by this equation. We have two unknowns. We have the thermal equilibrium magnetization M0 and our relaxation rate R1. So we have two unknowns. We can solve this with as little as 2 images. And again, it's good because we could potentially standardize it across sites. So for this study design, it was a longitudinal exercise study with an exercise group of 21 subjects and a weighted control group of 22. This was done in an elderly population with a mean age of 74. And they participated in a 12-week cycling program where they had 30-minute sessions 3 times a week. And for the imaging portion, how we got our R1 maps was variable flip angle T1 mapping. So this is simplistically put a 2-point fit to the T1 recovery or R1 rate. So helping our statistics, we did an ROI analysis here, looking at the dorsal medial motor ROI. So this is known very well to have the output for leg motor neurons. So anything going on in this region is outputting directly to the legs and helping you move. And as a reference ROI, we looked at the ventral motor ROI, which should have a similar cortical composition. It's still a motor region, but this time the output is to the head. So if they're doing a cycling exercise, we're expecting this region to be highly active during these sessions, something that wouldn't have been active before if they weren't exercising. And then we're not really expecting any change to the head outputs, at least in terms of activity. So if we look at that, we have the yellow arrow pointing here is the ventral region. So that's where the head outputs are, and the leg outputs are right in the middle center of the brain. So as we hypothesized, we saw an increase in the change in R1. So this is we're proposing to be an increase in myelination over the study in the exercise group compared to the controls over this range from doing this exercise study over 12 weeks. And we saw no change in the reference ROI, that was further down. So again, we want to know, great, we changed the structure of the brain. Did it impact the function at all? So we also had scores of their functional ability. So what was collected was called the VT1 or ventilatory threshold 1. This is sort of if you're increasing your walking pace from a slow walk to a brisk walk, you'll start to pick up your breathing rate at some point along that gradient. That's sort of your VT1. It's a very low end aerobic ability. But we found the change in VT1 over this study period between the exercise and control was correlated with a change in R1. So increasing their R1 in their motor cortex was associated with a better fitness score at the end of it. So that was really interesting. So with the R1 increases in motor cortex with exercise, we found increased physical fitness that correlated with a marker for intracortical myelin. And what's awesome is it shows cortical plasticity in the elderly. So it's never too late to start your exercise program for your brain health. So we'll see if we can lose people in this section. So what my work really is focusing on now is trying to develop a more specific myelin marker in the brain. So R1's a bit better than T1 divided by T2, but we're still sensitive to magnetic ions, manganese, iron. But to get something more specific, we'll have to exploit some properties of cellular structures to increase the signal specificity. And we're going about this by using MT imaging. So again, a quick refresher. The way we're using MT is an off-resonance RF pulse to selectively saturate the bound pool, and we're visualizing it through its exchange with the free water pool. So MT, it's dependent on numerous parameters. So here we're modeling it with a 3-pool model. So we have our free water pool, which exchanges with the bound pool. Each of these pools relaxes back towards its thermal equilibrium with some time constant. And it's saturated with some R constant R, RFA, RFB. And then we're also added a dipolar pool. So the dipolar pool is primarily carbon atoms with 2 hydrogens attached to them. And then those hydrogen protons have some dipolar order. We can model this nice diagram here with a series of coupled equations. If we didn't have power constraints, we could set these equal to 0 and solve them. But unfortunately, in human imaging, we're kind of stuck with the equations as is and have to numerically simulate them. So how can we use this information or model to develop a more myelin-specific contrast? So we know that myelin contains a very large dipolar pool. We have long lipid chains, and those lipid chains are just carbon, carbon, carbon with 2 hydrogens attached. That's not really the case with proteins. From NMR work, we know that using an off-resonance pulse with only a positive or a negative, so a single frequency that's applied with the RF pulse, only partially saturates the dipolar pool. So that affects this RRFD term. So how much of this dipolar pool are we saturating? If we're only using one frequency, we're only getting so far down. If we apply it simultaneously at both positive and negative frequencies, so if we were at positive 7 kilohertz here, now we're simultaneously saturating plus and minus 7 kilohertz, we can completely saturate this dipolar pool. So we're getting increased saturation of the semi-solid pool, which will show up as a even further decrease in free water signal. So if we take the difference of these two, if everything else is kept the same based on our imaging and we're looking at the same sample, so all the tissue parameters stay the same, the difference between the two images will really just report the impact of this RRFD term and give us insight into the dipolar pool. And in doing so, it removes the contributions from proteins and magnetic ions like iron. So some initial work was done to validate this by Varma in 2015. So the first thing was just in T2. So we have row 1, which is ovalbumin, which is an egg white protein. Row 2 is hair conditioner, which makes these cool lamellar structures, which is highly similar to myelin. We have these long lipid chains. And then rows 3 to 10 was just mixed concentrations of manganese chloride to alter the T2 value. So we see altered T2 just as a way to check for direct saturation with the off resonance. So if we look at the MT effect, because this is just water and ions, we're not expecting any MT. And that's what we see. 3 to 10 for MT effect is effectively 0. And then for the proteins, ovalbumin, we're seeing still MT effect, and also for the hair conditioner. So the more specific one was to take the dual minus the single, which they call IHMT, which is inhomogeneous magnetization transfer. And with that, you've now removed the contribution of protein to the MR signal, and you only have left this lamellar structure. So there's a growing field that's using this to look at myelin in the brain. So how do we go about doing this? A more simplistic sequence diagram. So a TR is a repetition. And for this simplistic sequence, we have one saturation pulse being applied. So that's the selective saturation of the bound pool, which is followed by a RF pulse to saturate the water. And this is where we're going to collect the imaging data from this pulse. So if this is repeated every TR, if we acquire a T1 map or an R1 map, then we know the relaxation constants and we know the thermal equilibrium magnetization of the voxel. So we know everything, and we can calculate the percent drop in signal that's achieved by modeling this signal as a steady state. So this point will equal this point. So if we do that, we can make maps across the brain. So here's a dual frequency saturation map and a single frequency saturation map. Again, all the imaging parameters were kept the same between the two. We've put the same amount of power into the saturation pulses. The only difference is that this one was applied at plus and minus 7 kilohertz, and this one was only plus 7 kilohertz. So the increase in saturation is solely due to the increased saturation of the dipolar pool. So if we want to take this and make an IHMT sat map, we can just subtract one from the other, and this gives us an inference into the dipolar pool. So now this map is more specific to the myelin content, as we've removed the contribution from proteins and iron. So unfortunately, this doesn't come free. So increased specificity generally comes at the cost of SNR. So this map's definitely a lot noisier than these two. So a lot of the work that's going on now that in our lab is trying to optimize this to see if we can improve the SNR in this image. So I showed you a more basic sequence diagram. What we're actually playing with now is something with a lot more parameters that can be tuned. So an issue in putting someone in the scanners, you don't want to be there forever. So we're trying to increase the turbo factor of the number of data lines we can acquire per time TR. We're also limited by how much power we can put into a subject. You don't want to fry them, but there's tons of safety regulations. So we're limited at some certain power intervals. So you have some dead time where you have to wait it out. You have the length of the pulse, gaps between the pulse. And then what seems to be a good way to generate this contrast is to have short little bursts, wait a time, and another short little burst. So we're sort of working with simulations currently to try to optimize this to increase the SNR, which will allow us to increase the resolution. Again, the goal of my work is to look at the cortical microstructures. So the gray matter is about 2 millimeters wide. So if we can't get to within at least a millimeter resolution, it's pretty hard to really look at this brain region without suffering from partial volume. I'll try to get through this pretty quickly. Just some work that I'm also working on right now, some biophysical modeling. So the whole point of trying to get more specific metrics is so that we can make inferences of what's going on at the microstructural level. So one of the ways to do this is to look at the G ratio in the brain. So the G ratio quantifies the thickness of the myelin sheath in relationship to the axon it's ensheathing. It's the radius of the axon, so this is the blue, divided by the complete radius of the axon plus the myelin sheath. This is an electron micrograph to show you what that looks like in practice. For MRI, we can do this on a volumetric level. If you go through the equations with an estimate of the myelin volume fraction and the axon volume fraction. So my IHMT work's really trying to solve this part. I didn't present work here today, but we're also working on some diffusion imaging to get a more accurate representation of the axon volume fraction as well. And the reason why we'd want to do this is to be able to differentiate different cases where you might have similar axon values, such as changes with axons with increased activation and learning. So the axon stays the same, but increasing myelination. But cases in neurodegeneration and demyelination, such as in MS, where you might have an initial demyelination, you want to be able to disentangle that from axon degradation. So that's something that's really motivating this work. And some initial cool work by Sean Dioni's group that shows the G ratio in action. So we can argue about the specificity of the metrics they've used, but I think this was some really cool work that show the myelination of the developing human brain. So this is from about 3 months of age. We have a pretty bright value. Again, the G ratio metric is inversely correlated with myelin. So a value of 1 gives you a relative myelin thickness of essentially 0. And then as you're going through aging, so now you're almost a year old, you're starting to get some contrast between the white matter and gray matter. And there's still continued myelination to 7.4 years. The color scale they use, it's pretty hard to see, but here you're hitting values of 0.8, 0.85. It's generally thought that the energetic ideal value is around 0.7. So I mean, this agrees with the work we showed earlier, where myelination continues to go up till 35, but it's really cool to see this initial rapid myelination of the brain in the first years of life. So again, my work's looking to validate this. So in order to generate specific markers, we need to be able to validate that it works. So we're using the Common Marmoset as it provides a simplistic model of the human cortex. It's great because it has this very smooth brain in relationship to the human brain, which helps us with partial voluming and curvature issues. And we're going to use this over something like a rat or a mouse brain because it has similar cortical organization and myelination patterns, which will really help with the translation from marmoset to humans. And I mean, the brain is vastly smaller than a human brain, but with specific hardware, we're able to capture really nice high quality images in the marmoset brain that will allow this work to happen. Really quickly, some just a very initial work, sort of comparing the use of different myelin metrics. So I've showed you today that we can get a bunch of metrics that all correlate with myelin content. But once we get down to increased modeling, the specificity starts to matter a little bit more. So something like T1 divided by T2 weighted ratio, once we've gone through the myelin volume fraction mapping, axon volume fraction mapping, and combining it to a G ratio, we start to get quite high values. This has been thresholded to only show the white matter. So we're expecting values, again, around 0.7 here. And it's sort of a weird pattern going on. We have values that are quite high, compared to something like IHMT-SAT, where we know it's a much more specific metric and the values are much lower in comparison. So we're working to follow this up with histology to validate this and to hopefully push it into the cortex where the microstructure is a little bit more complex with cell bodies and more proteins. So the overall conclusions from the work I've shared today: MRI provides a means to study the relationship between brain structure and function. And the intracortical signal changes can track aging and disease progression. And these changes correlate with functional scores, which really help us study brain plasticity and development. And the development of specific imaging biomarkers and biophysical models will ultimately provide enhanced microstructural information, allowing us to understand brain plasticity even more. So I'd like to thank all my collaborators at McGill, Calgary, Lund University, and McMaster for helping make this